now we talk to Tammy, who can, I'm going to give you an introduction and you can expand as much as you like. So Professor Robinson Smythe is from Stellenbosch University, where she holds a research chair in the management of invasions in protected areas. And she has a background in marine ecology, and as part of our workshop, of course, and has worked on the spread, impacts, and management of invasions. Particularly interested in the nexus between climate change and invasions. Let's see, what else should I say? That's maybe enough, right?
That's enough.
You can expand a bit on yourself as you talk. Yeah.
Thanks, John.
Sarah, welcome.
Thanks all for coming down today to have a listen, and I hope that I can share some interesting info about the work that we've been doing in South Africa. So just to point out that I've held this chair for almost 2 years, so a lot of the work that I'm presenting today it's really just starting to come through. A lot of it is not published yet or is in review. So while there's lots of other stuff that's been happening in my lab, what I'm focusing on today is really the work that we've been doing in support of management within South African MPAs. This way. Okay, so marine protected areas are established for a variety of reasons, and I think those reasons have really progressed and changed through time. And primarily, historically, they've been set up to protect biodiversity. But particularly in developing nations, they have other mandates as well, which include things like feeding into economic sustainability. So in a South African context, for example, there are MPAs whose primary aim is to actually protect fished resources. With the overflow effect, we can have discussions about whether that actually functions or not. But there are areas where MPAs aim to support needs of people and at the same time are able to protect biodiversity. The other thing that they do is they have a mandate to address human health and well-being, and this is becoming particularly important as communities are expanding, as urban areas are growing, people are needing places to get away to, and in a South African context, the focus on human health and well-being and that connection with nature is becoming increasingly important. So our MPA network has these 3 facets. And although as ecologists we like to focus on the biodiversity, we can't ignore the fact that there are these other aspects to MPAs as well. So what do MPAs look like in South Africa? I'm pleased to say we have a really decent MPA network. How it's come about has been quite interesting. So we've had some primary, that there is Tsitsikamma National Park. And that MPA is actually the oldest formerly protected area, terrestrial or marine, in Africa. We had a few key places like that, but we didn't have this amazing network. And it was one of the things which our ecologists and our conservationists have been working towards. But declaring MPAs is really a political issue, and we somehow ended up with a really interesting gap where our president decided that one of the ways that we could foster economic development within South Africa was to tap into something called the blue economy. So he was really keen to foster use of offshore oil and gas, fisheries, aquaculture. That was kind of the selling point of a political campaign. And this was really great because this gave a gap to negotiate marine spatial planning, and as part of that, MPAs were declared. So we went from roughly 10 MPAs to 41 MPAs. And I would say overnight, because the signature happened and suddenly we had 41 MPAs, but it was a lot of blood, sweat, and tears by many marine scientists that actually drove this. So now we have this really nice network. We have coastal MPAs. We also have offshore MPAs. Let me rather use these. So these ones over here are set up primarily to protect from fishing, particularly trawling, while some of our west coast MPAs, like this one over here, this is in a diamond mining area, and so offers protection against, in terms of extraction. And then we've got all of our really small MPAs in the West Coast region over here. Table Mountain National Park covers almost the entire Cape Peninsula, and we'll focus in on that MPA in a little while. So at the moment, we have coverage of about 5% of the country's oceans. As part of our commitments under the CBD, that needs to increase over the next while. So at the moment, there are negotiations already happening about extending our MPA network. So let's jump into marine invasions. As ecologists, we tend to focus on the ecological impacts, and those can be anything from genetic in terms of hybridization, right the way through to changing population size, extending through to ecosystem functioning. But there are also impacts for people, and those impacts can show themselves in terms of affecting economies, reducing fisheries takes, altering ecosystem services. We can also have impacts associated with human health. And here in the marine context, we primarily see this coming through in the terms of organisms that are introduced that result in bites and stings, but we also have ascidians that actually cause respiratory allergies. And there are a couple of people who are known to have died from mucking around in rock pools that had a particular ascidian in them. And then, lo and behold, they affect well-being as well. And I know that this is not an impact that we see everywhere in the world, but in the South African context, there's been a lot of concern around the traditional impact of invasive species on traditional ways of being. So young girls spend time with their grandmothers harvesting mussels on the shore. And grandmas take their granddaughters down, and it's not just knowledge about the environment that's passed on during that time, but there's social knowledge and family knowledge which is passed on through those interactions. And as alien species change those systems, the species that they go down to collect are no longer there, and so we're seeing decoupling of these social connections. So you might have before we jump onto that. So invasions are increasing, there's no doubt about that. And I've just taken one example here from a recent Sebens paper, which looks at fish, and we have the breakdown by just different geographic regions. What's very clear is that invasions are increasing through time. So we can't ignore them. And I'm sure by now you've seen that the imperatives of MPAs, which are to protect biodiversity, support economy, and protect human health and well-being, those imperatives can be directly undermined by the impacts associated with alien species. So if we don't manage invasions, we're undermining the ability of MPAs to meet their mandates. So this is not just something that invasion biologists should be concerned about, but really, if we're serious about conservation, we need to be addressing invasions in MPAs. So what do we know about invasions in South African MPAs? And this is where I start to cringe, because we don't know that much. The first dedicated survey only took place in 2003, and that was in an MPA called Langebaan Lagoon and an associated island, Marcus Island. A lot of work had happened in these areas before, but there hadn't been dedicated surveys looking for alien species. In 2010, a student focused in on the Betty's Bay MPA, and in 2015, one of my master's students got my dream project. He basically got to visit every single MPA around the coast, and he surveyed for alien species. And this is what he found. So for the first time, we had clear numbers of alien species present. There were only two which had no invasions. 16 Mile Beach and Helderberg are actually only sandy beaches. So everywhere where there was hard substrate, we had alien species. But we can't ignore the fact that ecological work had been happening all the time. And so we had detection of alien species, but they were really coincidental. It was while people were working on other questions and they'd come across an alien species. The most recent detection has been in Langebaan Lagoon in December 2023. I happened to be managing a paper. I was editing a paper for a journal, and they were talking about a soft bryozoan that had this global distribution, and I thought, how can this be global, and we've never seen it? looked it up, sent out a photo to all of my students and said, please keep your eyes open because this just doesn't make sense that we don't have it. And lo and behold, one of my PhDs went on holiday and sent me photos about 2 days later and said, I think it's here. And it was in the MPA that we found it for the first time. We'll get into the species a little bit more in a little while. Right, so one of the things I want to point out is that if you look at the numbers of species present in MPAs, we're not even hitting 10 in Ben's work that took place in 2015. Those numbers are really low, especially when you think about the fact that South Africa has 98 alien species that are on our official list. So there's definitely a lack of knowledge about what's going on in MPAs. We definitely don't have skills within our conservation agencies to address invasions. Our rangers are trained in a terrestrial setting, and it's those that have an interest in marine that end up working in MPAs, and then they learn on the go. And we certainly have limited resources to address MPAs. So this is really where I came in. This is the kind of setting that my work in MPAs started in. And I think we need to start just by thinking about that real need for cooperation between scientists and managers, because evidence-based interventions are going to be key to making a difference. So we started off by doing a survey, and it wasn't just MPAs, it was across terrestrial systems as well. And overarchingly, if you ask managers what they think about scientists, they're distrustful. They just straight are distrustful. And they feel that researchers don't understand the situation on the ground, and that we're bossy and we come in there, know it all, and we tell them how to do their jobs when they've been in the field for many, many years, and they have these youngins coming in and telling them what to do. They don't like researchers and they don't like scientists. If you talk to scientists, managers are lazy. They don't understand the theory and what's wrong with them because I published the paper, the information is there, why are they not using it? And so there's a real disconnect between these two groups. So it became very clear very early on that if we were going to do anything meaningful, we were really addressing a people problem, and then we were addressing an ecological problem. So this was our starting point. So talking to scientists, when they observe a problem, we do what scientists do best. We follow the scientific process, we ask the question, we collect the data, we analyze it. We're measured in terms of papers, so the paper goes out and we leave it there for managers to pick up and use. Managers do things very differently. If they observe a problem, they address it based on their own experience. What have they done before? What's worked for them before? Sometimes they'll talk to other managers and get a little bit of input, and then they'll go back and readdress if necessary. But it's really scary how these two never interact. So the very first thing that we started off doing was actually running a workshop with SANParks Rangers. So SANParks is our national conservation agency. And we knew very carefully, or we need to be very careful and not barge in there as scientists ready to tell them what to do. It was very difficult to get them to agree to come to start off with. And so we really started it as a conversation, asking them what they knew about invasions. Some of them didn't realize that that could even happen in a marine context. They knew about pines and eucalypts, but they didn't realize that that could happen in a marine setting as well. We really started to unpack their experiences in the field, and we wanted to know where we could help them. And once we got that conversation going, then we started to share information about things like introduction pathways and impacts that alien species have in the marine context. We ran these pictures over here are showing you some work that we did helping with identifying common alien species that they might come across in the protected areas that they work in. And we started to share why we are concerned about invasions in protected areas. And this man up here, whose name is Johan, is just such a lovely example, because when he walked in, he stood right at the back. He wouldn't sit down. He stood at the back and he crossed his arms. And he was so resistant. And eventually he sat down. And eventually I noticed the arms relaxed. And by the time we got him in front of organisms and on a microscope, he'd started asking questions, and those walls had come down. And by the end, he actually came and said, this was really, really cool. Thank you for including us. So it just shows that with a little bit of time and effort, we can bridge that gap between researchers and scientists. But I am a researcher, so I couldn't let an opportunity to gather data disappear. And so we ran some surveys with the rangers before and after the work that we did with them. And I'm not going to bore you with all of the social science side of things, but I did want to cherry pick this one bit of info. So this graph shows the number of people that answered yes to the question of, do you need more information on alien species in your protected area? And we asked them before the workshop and after. And very clearly there'd been a shift. They thought they knew what they needed, or they thought they didn't need to know much in the beginning. And by the time we'd finished, they were asking us for more information. So this gave us the clear evidence about what was needed. And it also meant that they were likely to be open to us starting to work in their MPAs and starting to get involved. The other really important thing that came out of these workshops was a personal connection. The researchers, well, we felt that we knew who we were dealing with in each of those MPAs, and those rangers now knew us by name. They have my email address. They can contact me directly, and that open communication is really key to making a difference. So if we are needing data and we're in an area where we really don't have the money to spend on collecting that data, how do we do it? And this is where we started to work on citizen science. And I just want to point out that this work is really driven by Sarah Ackland, who's a PhD student in my lab. The citizen science or community science data is really data which the everyday person out there is collecting and feeding into a system. It enables the collection of really large amounts of data. It's generally open access. And one of the best things about it is that it gives us data at really broad temporal and spatial scales, much broader than what we as researchers can go out and collect. iNaturalist is one of the databases or the platforms which are most commonly used. I know that it's used here in the States. It's used in many places in the world. And the way that it works, most people use their phones, but you can also, if you're diving, you can take photos using an underwater camera and then load them afterwards. So you take the photo of the specimen that you're looking at, you post it, and if you don't give it a name, it's classified as a casual observation. If you give it a name, it's considered as needing ID. So somebody else in the iNaturalist community needs to confirm that ID or say, no, you don't know what you're doing, maybe it's this species. And something which is research grade has been kind of ticked box twice with at least 2 people agreeing that that is the right ID. So the idea was really that it's self-perpetuating and that the community can help to moderate the data quality. And I do want to point out at this point that iNat data feeds into really big data resources like GBIF. So GBIF downloads anything which is research grade is able to feed into GBIF. And so GBIF routinely loads iNat data. So this is an example of what a record might look like. This is Caprella mutica, the Japanese shrimp. This was taken by one of our students. These records are really nice because they are time-stamped. It gives you a photo so you can see exactly what the person saw. It gives you the geographic information because they're geolocated through phones. There is an angle of taxonomic ID, although we need to remember that that is all done by the community. And then there can be a variety of associated descriptions. So that sounds great. What could possibly be wrong with that? There can be lots wrong with that. And there's real concerns around data quality, reliability, and accuracy. And for a long time, people have kind of poo-pooed the idea that there's a problem here because it's going into GBIF, and GBIF is amazing. Yeah, GBIF is amazing, but it doesn't mean that all of the data that they take in is actually great. So we were keen on looking at iNat data as a potential source of information on invasions in South African MPAs but we were worried about this problem. So what we've done is develop a confidence score. And this really considers 3 sources of uncertainty that may be embedded in any record. The first relates to the species, and it's really just a decision tree. There's a variety of questions, and depending on what the answer is, you may need to stop because you actually can't assess the confidence for that particular record, or you get a score. So for instance, if the species identification is possible with phenotypic features only, we can move on. Can you see those diagnostic features in the photograph that you're looking at? If the answer was no, you'd get a low score. If the answer was yes, you'd get a high score. So you can work your way through all of these. The next aspect of uncertainty relates to media. So these are all examples of photographs that had been listed as Mittelschala provincialis, which is an alien mussel in South Africa. The top 2, you can clearly see what the species is. We can tell that those are right. But these ones at the bottom, you've got no idea. We're guessing in the bottom right that what they're talking about is these over here, and the fact that we have multiple mussel species in South Africa tells me I don't really know what that photograph is of. So we can think about uncertainty in terms of the media that's been posted as well. And then we can think about it in terms of georeferencing. So if we're going to use this for monitoring, for example, we need to know how accurate the geopositioning is of that record. And when you take photos using the phone, it automatically records an uncertainty around that geolocation. And so we have, at this point, settled on a positional accuracy of less than 1 kilometer. We can chat about that if anybody's interested. But what it does is write off any of these crazy examples. Like this is a plant. There aren't any marine examples that are quite this extreme, which is why I put a plant example in here. But this is a terrestrial plant in the middle of a bay. Clearly nonsense. Okay, so it's one thing to develop a scoring system, but we needed to test it. So we used the NIS and cryptogenic species in South Africa. We searched for each one by name on iNaturalist. And then 2 people verified the IDs that had been allocated on iNaturalist. And that meant we were able to contrast accuracy and quality grade. Remember, quality grade is embedded in iNat, and then the confidence score that we developed. Okay, and I do want to apologize because when I put my slides up, I realized somehow the x-axis label for the top graph had disappeared. So that's actually confidence. So if we have a look at the top graph, we're looking at accuracy. So were the records correctly or accurately identified versus the confidence along the x-axis? And we can see that there's a significant relationship here. If we contrast accuracy and quality grade, there's also a significant relationship, but that is a weaker relationship. And I do want to just draw your attention to these. So if we're looking at confidence and we're looking at medium and high confidence records, we only have 81 records that we would end up including that might be incorrect or uncertain, okay? Whereas if we use the quality grade, which is the information which is currently going into GBIF, here they would have included 167 records which are really uncertain. It generally means you need genetics to ID these species, and 71 that were clearly wrong. So the community reinforcement of IDs had really fallen apart there. So the recommendation that's come out of this is really that we should be working with records which have a high and medium confidence score. And this work, while I was sitting in the room down the passage yesterday, was accepted for publication in Ecology and Evolution. So we're pleased that it's not just us that's thinking that this is a decent way of doing things. But the reality is that this can be a lot of work for an individual to sit down and do. So at the moment, we have a computer science student that is looking at automating this process, and it'll feed directly into Sandparks' database. So they're going to go from very little data to having an automated process which will at least give them some information on invasions. So we then used this approach and we applied it to South African MPAs. And that meant looking for all of those species within those particular areas. What this did was jump up the number of alien species that we know from 22 to 30. But we also had some really interesting findings. So this is a nudibranch that had previously only been recorded as once. And the question is always, if you find something once, has it established? Should it be on your list? Shouldn't it? We actually now know that this particular nudie is well established within South Africa, and somehow scientists had just not been picking it up. But when we had thousands of people out there working with INATs, we found it. Canaria had last been recorded in 1975. We now know that it's all over South Africa and it's still there. There are a couple of ascidians, Siona robusta, Clavellina, and Stela plicata, which we only know of from harbors, but now we're starting to pick them up in natural habitats as well. And then a couple of species that have now been recorded in different ecoregions. So our ecoregions on this map are just coded in different colors. So for instance, if one of them had only been recorded on our cooler west coast, it's now being picked up in warmer ecoregions. So we're starting to see really interesting patterns of species as well. So having worked in South African MPAs, we then decided to step this up and have a look at what it could tell us about African MPAs. We used Protected Planet to identify 79 African marine protected areas across 14 countries. So these are all of the officially declared MPAs. We used worms as well as literature searching to look for what species have been reported as alien in any of the coastal countries in Africa. And then we ran that entire list through INAT, and using the georeferenced layers from Protected Planet, we could actually use the INAT georeferencing and see exactly where each one of those records fell. So we knew which records were in MPAs and which ones weren't. We ended up with 422 records in total. So this is really the workflow that we developed, which we've applied to MPAs, but could really be applied to any particular area of interest. It's really just about identifying your area, being able to draw on shapefiles, compile the species list. In our instance, what we did was we drew in that info from INAT, but then we obviously had to double check maybe species that had been reported as alien in South Africa were actually native to Morocco. So we had to go through that whole data cleaning process. This is what we came up with in the end. So 21 MPAs in 10 countries, and these are the MPAs which had alien species in it. But I'm going to jump back for a second. So for now, I'd just like you to ignore the colors. We're just looking at numbers of species on the y-axis. The first part of our x-axis is all South African MPAs, and it's not surprising that we've got pretty good data there because there's people working there. But what we were surprised to find is that we had actually relatively high numbers in Mauritania. And when we did a little bit of digging, what we found was that there was actually a PhD student who lived just on the edge of the MPA and had been loading INAT records through 2020 when he was homebound and couldn't get out. So it shows you how important individual people can be in gathering this type of data. But also, if we engaged, for instance, dive companies, and we had buy-in from that kind of tourism sector, we could really start to bolster the information that we're getting. So I'm by no means saying that we have, an awesome complete data set here. What we have is a first step, and I think a proof of concept. What was valuable about this information is that 9 species were actually recorded in countries where they hadn't been reported from before in the literature. And this, I think, is really valuable because now researchers or conservation agencies in those countries can go and ground truth those records. We know exactly where that record was taken, where it was found, and now we can go hunting for, are these species still present? Is there establishment? Okay, back to the colors. So some of you might know about the IUCN endorsed ICAT and SciCat schemes. These are very controversial in the marine realm because we all think that they're a little bit of nonsense. But the terrestrial people have pushed them really hard, and they were developed primarily by botanists, and it's an approach to assessing impact by alien species. There is very little impact information that is out there on marine alien species. It's not just an African problem, it's a problem across the board. So when Sarah wanted to look at impact, I said, well, if you're going to do it, you need to use the IUCN gold standard, but I don't think you're going to get anything. And I don't know if I'm the only supervisor that has said that, and then they've proved me wrong. But Sarah went, and all of these colors are actually reflective of species whose impact she could assess based on reports in the literature. And the white ones are the data deficient. So I'd expected most of the species to be data deficient. I didn't think she was going to find anything. Just to point out that it doesn't mean that these species are having these impacts in these MPAs, but that these impacts have been reported for these species somewhere in the world before. So it does mean that managers and rangers can now be on the lookout for those species and can be thinking about what impacts, if they're not manifesting already, may in fact start to show. Okay, so overall this gave us our first list of alien species. In African MPAs, we had first records in particular countries, and I think the most important to me is really that it's given a foundation against which managers can start to prioritize which MPAs need to be monitored, what species do they need to be looking out for. And this can really help to support states in terms of meeting their obligations under the Kunming-Montreal Global Biodiversity Framework targets. I know the US is not part of that agreement, but most African countries are. And these targets have really replaced the old Aichi targets. So those have phased out, and these are the new targets. And target 6 particularly addresses invasions, and they are targets that particularly address protected areas and the need to minimize impacts on native species. So this kind of work can be put together to help to address both of those targets. Okay, so next we moved on to working in kelp forests, and this work is primarily done by Clara Steyn, another PhD in my lab. So you guys know that the South African systems are or our West Coast system is very similar to the system which we have in California as well. So we have kelp beds which range all the way from the border with Namibia up over there, all the way down our West Coast. And relatively recently, there's been a new population of kelp that has established to the east. And there have been a number of species that are cold species that have actually come down the coast and have spread eastwards over here. So in many places in the world, they're tracking a spread of species responding to warming, and we actually have cooling, which is happening in that area. So we've had cold water species moving in, and we've had warm water species retracting up the east coast. So a bit of a different setting to what you see in most places in the world. When Clara started her PhD, there had been no dedicated surveys for alien species and kelp systems at all. There had been 4 non-Indigenous species documented, but those were coincidental to other work that was happening. So there's a very clear need for surveys and monitoring, and that's the work that Clara started to focus on in terms of her PhD. And very broadly speaking, her PhD is addressing what should we be looking for? Where should we be looking? How should we be surveying? And when should we be running these surveys? And I could fill an hour just with this, but what I'm going to do for now is just focus in on the what. So target lists are incredibly valuable when you're surveying for alien species, because it means that the people who are doing the surveys know what they're looking for, and they can be much more efficient in looking for those species. But the downside is that you can miss things that are not on your list. And that's really where watch list comes in. So watch lists are developed generally part of horizon scanning, looking for threats that may come through in time. Watch lists allow us to look at what species have become established or are alien in similar settings elsewhere in the world. So which ones have an invasion history? Could they get to South Africa? And then that would be one of the ways of establishing a watch list. So the way Clara did it was she surveyed the literature looking for species with invasion histories. She then assessed whether they had a pathway to South Africa. So could they cross that geographic barrier and get to South Africa? If they did, and that's where most people stop. But really, I think it's important to think about that physiological barrier as well. So if they arrive, but there's no way that they can survive in our conditions, well, we're not too worried about them. And I know that there are many aspects to physiology, and we couldn't possibly account for them all, but what we did do was look at temperature. So we looked at temperature matching. And through that whole process, what she was able to develop was a watch list for potential invaders in South African kelp beds. In terms of the, excuse me, in terms of the climates, making these lists climate sensitive, we looked at not only current conditions, but into the future as well. So we looked at warming using the IPCC predictions, and because our West Coast has a documented trend of cooling, we also looked at cooling. And although that's what we're seeing very often when we try to publish internationally, people go, these Africans, it's warming, it's not cooling. And then they give us trouble about looking at cooling and not warming. So now we simply look at warming and we look at cooling and we cover our bases. And we did this looking at a severe and a moderate scenario, and we also looked in the near and the far future. And I'm not going to get into the details of this, and we can use the colors to guide us, but essentially if a species, so we looked at the species thermal range in its realized range. Okay, so everywhere where the species is known, both alien and invaded ranges. And we said, we took the min and the max, and if that falls below and above the ecoregion, min and max in South Africa, that area is considered suitable. And we have increasingly, or if an area is increasingly too cold for that species, we see the cooler colors coming in, and if the area is too warm, we see the warmer colors coming in. So just to orientate you along the South African coast, we have a really interesting system, as our west coast is very similar to the Californian coast, cold, temperate upwelling. We have really warm system on our east coast, and then we have everything in between in the middle. And so we have very different ecoregions along our coast. Because we have kelp both on the west and the east coast, we needed to consider those two areas separately when we looked at temperature matching. So just as a reminder, that the west coast would be the southern Big Wela, and the Agulhas is on the east coast, the south coast. So this is the list that Clara developed. We have one bryozoan, Membraniptera membranacea, and then the rest are all algae. Right, so all of those have an invasion history elsewhere. They have pathways to get to South Africa, and here she's using the pathways as assigned by the CBD, so stowaway, contaminant, and escape. And let's just look at present at the moment, just remembering we're looking at the Agulhas region here. So none of the species are excluded from this region. They can all survive the thermal regime in present, although sargassum, it's a little bit cool for sargassum, and it's on the warmer side for the other species. But then looking to the right, regardless of whether we're looking at moderate or severe warming, we have pretty much the same thing going on also through time. In terms of cooling, if the Agulhas does cool, it becomes more suitable for Dasyphonia. Right, so no species are outrightly excluded by temperature, and I think that's really important. We have the same thing along the west coast, excepting that here it is cooler, and so it makes it more suitable. And this is not surprising because kelps occur in temperate settings, and so that this all makes sense. And one of the real kicks, I think, that scientists get is when you get proved right. And this work is currently in review. And in the work that Clara was doing, looking at the best way to survey for alien species in kelps, what did she find? She found Membraniptera. So we now know that actually it's arrived, even though we've only just put it onto our watch list. So I think these photos come from this region. At the moment, we've detected it 5 times in different places, but always at low density. It can be really devastating in kelp systems. So we are particularly worried about what's going to happen with Membraniptera in the South African context. But it's certainly one of those instances where if it's there and it's established, the horse is bolted. So I hope I don't come back in a few years and show you pictures that look similar to this. But we're yet to see what the impacts are in a South African setting. Okay, so if species do arrive and do establish, what do we have at our disposal? And this is some of the work that Miranda Anderson is doing as part of their MSc. Eradications in marine systems are notoriously difficult, and this graph gives us an idea of the area impacted by a species and how that changes through time. So initially, if the species is absent, obviously there's no area impact, but once it's been introduced, there's a lag phase before it starts to increase in distribution, and then area impacted goes with that. And the idea is really that if you can manage a species early on, where it's relatively confined, that's your best shot at doing anything about it. It's also the best time in terms of economics. So if you're working in a resource-constrained setting, you can't afford to try to deal with a marine alien species when it's widespread already. So the key here is really that rapid response is needed. But rapid response needs good monitoring because you need to pick it up really early on. And we're hoping that using INAT and having more eyes in the water, we're going to get to that point. But what happens if you've missed that point already? So generally, if you're going to mount a response, you're going to need three portions or 3 areas that need to be considered. You need to be thinking about what are you practically going to do? What are you going to do? How are you going to do it? When are you going to do it? And that's really where scientists generally come in. We also have implementation to think about, and in an MPA setting, this would generally be the responsibility of the rangers, and then we would have admin. And admin needs to make sure that the finances flow, that permitting flows, because without that structure there, nothing can happen on the ground. And yet these 3 portions are, or different people are responsible for these 3 portions. So in reality, it takes too long to get rapid response up and going when a new alien is detected. So if we recognize that we have alien species which are present, but they're only, for instance, in a harbor, they haven't spread that widely yet, we know we can't deal with them where they are, but we're very concerned about a particular MPA or a particular aquaculture area or a heritage site. We can think about developing rapid response plans for that particular area. So then you're monitoring not everywhere, but you're monitoring a specific place, and you have the time to develop the ideas about what you're going to do, who's going to do what, how are the finances going to flow, and you can be ready and actually be effective if you pick up that species in that particular area. So of course, we're thinking about that in terms of MPAs. And this is the essentially the workflow which we've developed with Miranda. And step 1, so I'm just going to focus on where there's solid lines to start with. So step 1 really is about filling context-specific knowledge gaps. And the blue area, let's start off over here. This is really when you're thinking about the biological traits of the species that you're interested in. What is the propagules pressure like? if you're going to be managing that species, you also need to be thinking in terms of the area of concern. What does that area look like? Are there pounding waves? What is that going to mean for having people in the field? What are the social dimensions around that area? Do you have local communities who are going to be very resistant to removal of an alien species or use of a particular method? So you really need to understand the gaps in knowledge which you have, and if you can ID those early on, then you have time to be able to go out and address them. So that's the first step. The second is using the literature to identify potential control methods. So once you know about the biology of the species, you can start to look at how have other people dealt with this in other places. And if no one's dealt with your species, how have they dealt with similar species? And then you can use all of that information together to identify potential control methods, which you now go out and test. Because we're not into gut feel work, we want evidence-based work happening. So steps 1, 2, and 3 should be done with researchers together with conservation agencies. The fourth step is then around assessing suitability of potential methods. And so this is really thinking about, would it be appropriate to use in an MPA, for instance? Maybe there's an awesome chemical that you could use to sort out the alien, but it may not be appropriate to use in that particular area. And then step 5 is really assessing feasibility. So this is where we start to think about the economics, and that comes down to the conservation agency. The added layer here is we may have a setting where we know the species we're concerned about, but we don't know where it might go to. And you cannot develop these plans for every single MPA. So if we have a new species of concern that arrives in a particular harbor, we may have 20 MPAs which are at risk. And those will have different social contexts, different physical traits, and it's just not feasible to develop these plans for everywhere. So you can develop things to the point of knowing what methods might be appropriate, but you can't fill in this section over here. So until you have all of the information, and be that, I don't know, SDMs potentially could give you that information. There's a variety of ways that you could fill that info in. And if you can't, at the moment, the thinking is that you should be able to get everything in place accepting that. And so even if you pick up that species in a new area, you should then be able to fill this in very quickly. And it means that you start over here, you don't start at the beginning, which speeds everything up. So Miranda is running a test case using an alien anemone in Langebaan MPA. Celeste ornata was first ID'd in South Africa in 2001. Since then, its ID has been called into question, and so at the moment she's busy with genetics and histology because we think it's actually got a different name. So for now, we're calling it Celesta spa, or Celesta South Africa. It occurs in the lagoon, both on fossilized oyster beds and in between seagrass beds. And the problem here is that seagrass is locally threatened. Locally endangered. It's threatened at the scale of the country. And so really Miranda's work is first confirming that the idea of that species. She's going to be doing ensemble niche modeling to try to identify other places where this alien may spread to. I do want to point out that it hasn't gone past us, that it's been around for so long and it hasn't spread. Yes, I know that, but that's really why this is almost a good test case, because we have the time to develop the ideas of what we would do should it be found somewhere else. Okay, so in terms of step 1, it's really around filling the knowledge gaps. So here she's been searching the literature for other alien anemones. What are the biological traits around this particular anemone? For instance, it has live young. So whenever you pick them up, you squish them a little bit and out pop all the babies. So knowing something like that is really important, because if you're going to start removing them by hand, you're going to be squishing out little ones, and what are the implications of that? She's been looking at potential control methods. for a variety of soft-bodied organisms and benthic species. And we're now in the process of, well, she's screened those together with Stan Parks. We've ruled out things like chemical treatments and heat treatments, as well as biological control. So that we're now at the point of assessing suitability of potential methods. And this is where the science really comes in. So it's about developing strong experimental design to assess things like, if we're going to remove them manually, do they survive once we pull them out? If somebody drops one or two, are they going to survive once they've been loosened off? You've got to know what you're working with. Otherwise, you can launch a management intervention and then you can end up wasting your time totally. She's also going to be looking at the effectiveness of both manual removal as well as salinity control, so hypersaline and hyposaline. And then we're going to take those lab experiments and roll them out in the field and look at the effectiveness of actually removing the anemones off the oyster beds. At this stage, we're still in discussions with Stan Parks about the seagrass beds because it's endangered. They don't want us mucking around, understandably. But they, at this point, are in agreement that we would be able to manually remove off the seagrass, but they're not so keen on the use of salt. And then we're going to track the effect on the seagrass. And the aim really is to develop a draft response plan and hand that over to Stan Parks, who will then take it further. So one of the places that we are concerned about is Knysna Lagoon. Knysna Lagoon is very similar to Longabon Lagoon. So we have Longabon over here on the west coast, Knysna is on the south coast, but in geological time, there was actually a connection between these two, and we have some unusual warm water species that occur only in the lagoon and in Knysna. So it's a really interesting setting. So if something can live in Longabon, it can live in Knysna. And this is where work by Dan van Black comes in. So Knysna is really interesting, and I had to smile when I went to look at the redwoods before coming up here, because we have yellow woods, and our big forests are all in the Knysna area. The one thing we have that you don't have are ellies. We have forest elephants. What we also have, though, are increasing numbers of marinas, and there's a lot of pressure to increase the size of the Knysna marina. But Knysna is an MPA, and so what are the implications of that going to be if we're going to have more yachts coming in that could increase propagule pressure? So that's really the focus of Dan's work. And by now, it shouldn't be too shocking to you that there are no surveys for NIS in Knysna. There's no baseline information whatsoever, and we know we have this threat of increased propagule pressure. So it's really important that we get in there. We're going to tackle this from a variety of ways. Surveys of marinas that are present already, as well as natural habitats. Of course, we're going to tap into INAP and see what we can get out of that. We're using the CIRC protocol, so the work that the Smithsonian Institute does on settlement plates. We're using that protocol. And then together with Mark Rees, who was a postdoc here a number of years ago, we're actually going to be using eDNA, particularly looking at sediment. So Mark has some really cool info that's just coming out at the moment, where he's tracked invasions back over 200 years in Liverpool. They've got some really nice eDNA signals coming through there, and you can actually track the sequential arrival of species in that sediment. I'm not the hugest fan of eDNA, but he's convinced me to give this one a go, and particularly in terms of the sediments. I think it's going to be interesting to see, because that'll allow us to look back in time in Knysna, as well as get an idea of what is present at the moment, and then we'll be able to track going forward. The other work Dan's going to be doing is looking at predators as a source of biotic resistance. So fish and other macro predators are important or can be important in controlling marine systems. It's interesting, though, that meta-analyses have found a much weaker signal in aquatic systems than terrestrial systems. And I really wonder if that's because marine papers have often not taken into account things like tidal cycle or even nocturnal predators. And this is something that Dan's quite keen to do. We're going to be doing it using videos as well as plate, so there's a variety of approaches that we'll be applying. I think one thing that's really interesting is that in Brazil, they've found that pufferfish are really important predators in this context, and there's a pufferfish in Knysna that is seen as a pest. And people use it for bait for other fish, which I think is quite unusual for a pufferfish. But if we are able to identify predators that actually feed on fouling, it'll be really important in terms of potentially making sure that those predators are protected and not overfished within that MPA, because fishing is allowed in Knysna. And then the other thing he's looking at is using bubbles to control fouling on infrastructure and yachts. And this is by no means something novel. The guys in Australia have had some real promise using Knysna, and it could be a really helpful approach to use in sensitive areas like MPAs. So the last thing before we finish off, I just want to zoom round to Amathia. So Amathia verticulata is a really nice example of how those initial workshops that we had with rangers have really been so helpful. When Sarah first discovered Amathia, so this is actually the initial photo that she took of it. It's on a walk-on in Langebaan Lagoon. And it was the start of the December holidays, so that's our summer holidays. And we sent out an email and we said, we've just found it. We know you're all going on holiday, but we've just found it. And that was it. The rangers were saying, that's it, come, we've got you, let's go. And we were going, hold on, we've just finished, marking scripts and we still have to load marks and all sorts of things. They were so keen. They understood that if we get in there early, we can make a difference. And so, after confirming that this really is the species that we have, we went out and did some surveys. The full surveys happened just after I saw John, in fact, in January, and we were really horrified because so that's a clipboard over there, and this pile of brown is actually Amathia, which is now sitting on top of the green, which is our endangered seagrass. And so we did full surveys of the lagoon. The numbers 1 to 15 are different 3-kilometer areas, and we surveyed intertidally and subtidally. And this data is just colony counts. And where you see the numbers with the little seagrasses, the seagrasses indicate which areas have seagrass. And then where we've got numbers above the bars, those are colonies which were larger than a quarter meter squared. So really, really big colonies like that. And SANParks was keen to manage until they realized how widespread it was. And we said, look, although we thought we found it early, I don't think there's anything that we're going to be able to do here. I think you need to hold your horses. And because they were so super keen, we went out in the pouring winter rain and we resurveyed in winter, because the literature has suggested that it dies off in winter. And I just couldn't imagine how we could have such huge colonies and then them really disappear over winter. But that's exactly what happened. We actually couldn't detect it in winter, which has really given us insights into how we might manage it going forward. And I think the key management intervention here has to be prevention of spread to other MPAs and other lagoons in this area. So this is a work in progress, but a really clear sign of having the right relationships with conservation agencies can really make a difference. So finishing off, I just want to point out that my take-home message here is really that we need to focus on evidence-based conservation, and it's absolutely essential if we're going to minimize costs and be efficient and effective in managing invasive species. I think that developing good relationships is really, really key. And I'd also like to put out there to students in the audience particularly that you don't need to choose between good science and applied science. Those are not separate things. And with that, I'd just like to acknowledge all the hard work of all of my collaborators, and thank you all for listening to me today.
Aaron, can you go?
Like really small question. One is in the figure, where you calculate the thermal range, right?
Yeah.
So I want to know what is the, like, how do you measure species min and max? Is it the region where they come from? The,
Yeah. So there isn't the information. Nobody's actually tested thermal tolerance of these species, so we don't have that empirical data. So the best that we could do as a surrogate was to say, to take their realized range, so both native and invaded ranges, and look for the min and the max of those. I mean, and that is coarse, and there are issues with that, but that was the best that we could do.
Okay. And the curve where you have this time going and the growth of the species which are invading,
Yeah.
that was really like smooth. Can it be like sudden shoot up there?
Yeah.
How do you know at what time point you are currently at?
So that wasn't work that I did. That was work. I can send you the paper because that's global data. So it's a huge amount of data. Yeah.
Yeah.
Hi, this should be a problem. Gambling.
Talking to
I don't have gambling. I'm wondering how you're able to, what sort of metrics you use to determine success of management. I've mentioned several times looking at different methods, control these. Have you thought about how to evaluate success when stop don't have?
Yeah, so that's down the road for us. I mean, we've thought about that in terms of the Carcinus work that we did when we tried to, in vain, eradicate Carcinus from a particular harbor. And at this point, I'm not keen on management that's trying to clear things out that are well established. So when it comes to to clearing out a newly detected species, I think it's going to depend on things like the biology of that species, what density it was at to start with, and what that environment looks like. And I think as long as we get in there early on, we stand a decent chance, but those are the kinds of things that need to be embedded in that plan, because you can't put all this effort in and then not know how you're going to measure it. But I think it will depend on which species you're dealing with.
Yeah, super cool work with the managers. Two questions with that. So one, what is usually the background of the managers, both in terms of education or just population-wise?
So it depends where you are in South Africa. In the area that we're in, sure, that's an interesting one. Largely male, generally Afrikaans speaking. Education-wise, they have a diploma. So there is some post-matric or post-school education, but that is all focused on terrestrial systems and conservation. So they can ID trees and tell you how to conserve back. Like that's the kind of training that they have.
And when you said that they use their own experience to deal with a problem, like, could you give us an example? Like even with trees, like whatever they do, yeah.
So there's somebody who decided that, in his experience, pulling trees out and digging the roots out, there was, I'm not going to remember the name of the alien tree. He needed to dig the roots out to be able to remove that tree properly. And as scientists, we know that if you cut the stem at a particular place, that tree does not resprout. It's not a resprouter. And he thoroughly believed that the only way to get them out or to get rid of them was to dig the roots out. So they were investing a huge amount of time and energy removing every single part of a tree, including its roots. And it didn't matter what scientists said. That's what he thought he needed to do. So that's what he did.
Yeah, you mentioned that there's been 98 invasive species reported in South Africa, and I was wondering if you have a sense of how many of those are confined to harbors and estuaries, how many of those found outside on the open.
Very few are in estuaries. because our harbors aren't in estuaries. I would say 4 or 5 are in estuaries. Yeah. Open coast, if you'd asked me this a few years ago, I would have said 2 mussels and a barnacle, because they are very widely spread. But now that we're starting to get more info through INAT, we're realizing even the Scythians are actually on the open coast. So I don't have a decent number or a solid number to give you, but I think it's more than what we've thought of in the past.
Super interesting. With all of the data that you've been collecting, particularly with the INAT data that's geo-referenced, where you have the information within the MPAs, have you done any comparisons of invaders inside versus outside of MPAs in these different areas? And is there a signature of MPAs influencing the number of invaders?
We haven't analyzed that data yet. We do have it, and I think that's a really important question. We're starting to think around buffer zones, and especially because they're looking at establishing new MPAs, thinking about where those need to be, and distances from harbors, things like that is super important, but we haven't looked at that yet. Yeah.
Sure.
Hi, thanks for the talk. It was awesome. Thinking about the pessimist point of view here, I think what you say makes sense about not trying to do management actions tackling well-established invasives. From that pessimist point of view, if you look like 100, 200 years in the future and you have all these expensive plans to try to minimize spread, but things will like consistently or like once in a while slip through until it adds up and adds up and adds up. What's the pitch to managers and politicians to actually manage invasive species in that context?
So interesting that you put it that way around, because actually the pitch is a political one, because the impacts happen now, and so politicians have to answer to their constituents now. And that's why they're open to trying to address the problem now, even though you could argue that in 200 years' time, there's just going to be homogeneity across the board. Yeah, that's a red wine discussion.
Really interesting, thank you.
Cool.
Any more questions? I think we'll wrap it up. Thank you, Tammy, so much. Appreciate it.